The story of our life starts with just one tiny cell. As we grow up, the number of cells in our body increases to around 37 trillion cells. However, this process does not stop there. Our body must continuously generate new cells to replace the old and damaged ones. For instance, we shed about 50 million skin cells daily, and it is essential to replace these lost cells. Now, to produce new cells for both growth and repair, the DNA within the cell must first replicate. DNA is a molecule that holds the instruction for how cells grow and function. It is made up of 4 types of building blocks called nucleotides. Nucleotides are arranged in a specific sequence to form a long strand. Each DNA molecule consists of 2 such strands which are held together by weak hydrogen bonds, with A bonding with T and C bonding with G. This complementary bonding allows each strand to serve as a template for the synthesis of a new strand. This process is known as DNA replication. It is a carefully choreographed process that requires numerous enzymes and proteins, starting with the initiator protein. This protein is responsible for initiating the process of DNA replication. It binds to a specific location on the DNA called the origin of replication. Prokaryotic cells have a single origin of replication on their circular DNA, while eukaryotic cells have multiple origin of replications on their long linear DNA. When the initiator protein binds to the DNA, it causes a short segment of DNA to separate, exposing a small opening where an enzyme called helicase can attach. Helicase can separate the 2 strands of DNA by breaking the hydrogen bonds that holds them together. It binds to one of the strands and travels along the DNA molecule, continuing to separate the strands. Single-strand binding proteins, or SSBs, prevent the separated strands from rejoining by binding to both strands and preventing the formation of hydrogen bonds. When the DNA strands are separated, they can now be used as templates for the synthesis of new strands. This process is catalyzed by the enzyme DNA polymerase, which creates a phosphodiester bond between the 3' carbon of one nucleotide and the 5' carbon of the next. However, DNA polymerase cannot initiate this process on its own by joining 2 free nucleotides. It can only join a nucleotide to an already existing chain. This is where the enzyme DNA primase comes in. It creates a short RNA strand called a primer using the DNA as a template. This primer is used by DNA polymerase as a starting point for synthesizing the new strand. At the site of replication, the double helix structure of DNA changes to a new Y-shaped structure called a replication fork. Typically, DNA replication occurs in 2 directions, resulting in the formation of 2 such replication forks. As the fork moves forward, the unwinding of DNA causes a buildup of torsional strain ahead of it, resulting in the formation of supercoils. These supercoils must be removed for the replication fork to continue moving forward. Topoisomerase is an enzyme that performs this task. It binds to the DNA strand that is under tension and cleaves one of the strands. The other strand is then passed through the opening in the cleaved strand, effectively untwisting the DNA and reducing the supercoiling. Next, topoisomerase religates the broken phosphodiester bond and releases the DNA. This process is repeated multiple times to allow the DNA molecule to return to its relaxed state so that the replication fork can move forward. As the replication fork advances further, DNA polymerase adds nucleotides complementary to the template strand to the newly synthesized chain. Because the 2 DNA strands possess opposing polarity, replication must occur at the 5' end of one strand and at the 3' end of the other strand. However, DNA polymerase requires a free 3' hydroxyl group on the growing strand, thus synthesis can occur only in the 5' to 3' direction. This allows for continuous synthesis of one of the strands called a leading strand. In contrast, the other strand has to be synthesized in the opposite direction. Hence, this strand is synthesized in short fragments known as Okazaki fragments. Each Okazaki fragment has its own RNA primer, and DNA polymerase extends the chain till it encounters the RNA primer of the previously synthesized Okazaki fragment. Thus, the leading strand is replicated in the direction towards the replication fork, and the other strand, called the lagging strand, is replicated in a direction away from the replication fork. Eventually, the RNA primers are degraded by exonucleases, and this ensures that the daughter strands are completely made of deoxyribonucleotides. Once the RNA primers are removed, the DNA polymerase can finally extend the chain to complete replication. The remaining gap between the two fragments are sealed by an enzyme named ligase, which forms a phosphodiester bond between these two DNA fragments. DNA replication will continue forward till eventually one replication fork meets another replication fork. In circular DNA, this happens at the region opposite to the origin of replication. In linear DNA, this happens at multiple sites, and this marks the end of DNA replication. Once DNA is replicated, it can be partitioned into the newly dividing cells. And once this is done, DNA dictates the structure and function of the new cells by deciding which proteins have to be synthesized. I'll be explaining in my next video how this happens, so stay tuned. Thanks for staying till the end. If you like this video, do hit the like button and don't forget to subscribe for more videos like this.